I'd just like to check if Yep, it's working. Okay, I'll start. Hello, everyone. My name is Murtaza, and I'm a fifth-year PhD student at Brown. Today, I'll be presenting our work, single-cell GrapHiC, which is a deep learning-based approach that predicts single-cell Hi-C contact maps from single-cell RNA-seq. I'll start off my talk with a little bit of a background, although I anticipate that I don't need to go into too much detail. Anyways, so we sequenced the human genome back in 2003, and over 2 decades later, we are still in the process of unraveling how the same DNA sequence, which is shared by all the cells, lead to such differential, functional forms. A part of this mystery can be explained by how the same DNA folds onto itself to form this complex structure known as the chromatin. And it's well understood that this chromatin takes differential structure across different cells. And it's also fairly well investigated that small rearrangement in the structure, chromatin, can lead to differential gene expression. A key technique to study the structural heterogeneity in across different cell types is called Hi-C. Hi-C is a chromatin conformation capture experiment that takes in a tissue sample and produces a genome-wide two-dimensional contact map with n by n bins. Each bin in this contact map corresponds to a region of DNA, which is often referred to as a genomic loci. Each cell in this contact map counts how frequently two genomic loci were found to be interacting with each other in the 3D space. Analysis of these contact maps unveil crucial structural elements such as compartments and also TADs. And the problem with Hi-C is that it's very expensive to acquire. So what people have done in the past is that they've developed computational approaches to produce these Hi-C contact maps using a deep learning or a machine learning based approach using some cheaper or easier to acquire genomic measurement, such as these techniques often have relied on chromatin accessibility signals through ATAC-seq or DNS-seq, ChIP-seq, particularly for CTCF transcription factor, or histone modification ChIP-seq to predict these cell type specific Hi-C contact maps. However, things have changed recently. The recent advancements in the sequencing technology has brought about this single cell revolution. Single cell experiments, unlike bulk, unveil cell level heterogeneity, which is often masked away in the bulk sequencing. Specifically, the recent single cell version of Hi-C protocol highlights how genome organization differ. Genome organization differ at a cell level resolution in a tissue sample. While we know bulk Hi-C is very expensive, we anticipate the single cell version of Hi-C to be even more expensive because single cell protocol is substantially more experimentally challenging. So in the study, we asked the question, can we develop a computational method that can produce these single cell Hi-C contact maps? So to answer that question, we start off by looking at what do we need in the first place. So what we really need is to predict single cell Hi-C contact map is another genomic measurement. So in this case of single cell experiments, an experiment that measures two genomic measurements at the same time are called co-acid experiment. When we were designing the study, there were only two types of co-acid experiment. First one was first co-acid experiment measured DNA methylation with single cell Hi-C. The other one measured single cell RNA-seq with single cell Hi-C. We chose to work with the second one because single cell RNA-seq is substantially more commonly available. And hence, any method that we develop that can map single cell RNA-seq to single cell Hi-C would be significantly more useful. With that out of the way, the overall aim of this talk and this work is to build a method that we call single cell GrapHiC that is able to map single cell RNA-seq data to some single cell Hi-C contact map. In the next few slides, I'll briefly explain what are the challenges of working with such a data set and what do we need to do to accomplish this task. The first core challenge of comes from working with a single cell data set by themselves, which is that they tend to be very sparse. Single cell RNA-seq has around 90% missing values. And the sparsity is even worse in single cell Hi-C, where we typically only get around 5% of genome covered per cell. This challenge needs to be explicitly taken care of because deep learning or machine learning based models are notoriously bad at extracting any representations that can generalize to some other unseen task. To reduce this sparsity, what we do is we pseudo-bulk our single cell data based on their cell type annotations. What we do is we combine all of the single cells with the same cell type annotation into a single cell type specific data point. Through this pseudo-bulking process, what we do is we substantially reduce the sparsity while still preserving the cell type heterogeneity that we care about. However, we do give up the cell level resolution that we have in our data. The second core challenge of working with single cell RNA-seq comes from the fact that the gene expression is a consequence of complex interactions in the chromatin space, and hence is weakly correlated with the structure of the DNA. So essentially, what we need to do is come up with some augmentations with the single cell RNA-seq. So to augment the single cell RNA-seq data, we have three additional data modalities. The first one is the CTCF motif scores, which provides us with the location and the likelihood of where the CTCF protein will bind with the DNA. CTCF is well known to be one of the key actors in maintaining the chromatin structure. These scores have also been previously, these CTCF motif scores have been previously used to improve high-C prediction performance. The second thing that we gather are the CpG scores, which capture the GC content of a genomic loci. Studies have shown that regions of genome that has high CpG scores tend to be transcriptomically active. And since they are active regions, they can be used to infer the A compartments or the active regions of the genome. Lastly, to acquire additional structure, we propose a novel deconvolution step to extract relevant structure from an arbitrary bulk high-C to inform our cell type specific prediction. This extraction works on the insight that the higher order genome structure, such as larger TADs, are typically conserved across different cell types and hence can be extracted to inform our cell type specific predictions. The last major challenge of working with single cell RNA-seq is that single cell RNA-seq is inherently a sequential data, and it abstracts away the structural mechanics of chromatin. And so as a solution, we model our data as a graph that has genomic loci as nodes, and the high-C reads from the bulk high-C contact map serving as edges connecting those nodes together. Moreover, each node in this graph has single-cell RNA-seq expression, CpG scores, and CTCF motif scores serving as its features. This representation provides us the unique opportunity to naturally merge all of our data modalities in a single representation that does not enforce any strict geometrical constraint on our modeling. Moreover, in comparison to the existing approaches that model this prediction process strictly in a sequential way, we can embed this very important biological prior, which is that biological features that are closer to each other in the 3D space tend to be mechanistically or functionally related to each other. Okay. Now that we have gathered all the ingredients, now I'll discuss the architecture of how we bring all of them together to do our prediction how do we predict single-cell high-C from single-cell RNA-seq inputs. So in the next few slides, I'll briefly discuss individual components of our model. The first component is the positional encoding module. Its main task is to embed a positional prior with single-cell RNA-seq, CTCF, and CpG inputs. It does we acquire this positional prior from the bulk high-C by extracting eigenvectors of the Laplacian of the bulk high-C matrix. We only pick the top 16 component of this Laplacian from these eigenvectors because we do not want to bias our structural prior too far towards the bulk high-C since it can be chosen arbitrarily. These positional encodings essentially empower our model to learn features and extract representation that are conditional on how these features are positioned in the 3D space. The second component, node feature processor, extract local and global features in and between all the modalities in our feature set. It does so by first applying a 1D convolution block on each modality individually, which extract these local features, and then we pass on these local features to an encoder block with self-attention that will learn all the global interaction between all of the local features within the same input modality and across the modalities, conditional on their position on the genome. This module embeds the interaction between all of the modalities, conditional on their position in the chromatin in a shared representation space. The third component, the graph encoder, extract additional structure from the bulk high-C matrix. To achieve that, first thing that we do is that we merge the rich node feature set that we produced in the previous module with the bulk high-C to form our genome graph. Then it applies a graph attention operation on this genome graph to perform the deconvolution step. The attention mechanism in the graph attention operation does this deconvolution step essentially by assigning a low attention or a zero attention score to edges that are not structurally informative, um, essentially extracting only the structure that is relevant to do cell type specific prediction, while discarding rest of the structure it doesn't care about. The last component is the graph decoder that maps these node embeddings from the graph encoder to our final single cell high-C contact map. It does so by first taking an inner product between all of the node features to acquire a contact likelihood map, and then we apply a stacked 7 stacked residual convolution blocks to finally map this contact likelihood map to our final single cell high-C predicted contact map. Now that I have all of that out of the way, so how do we test and train that model? So we use the high-res data set, which is a single-cell RNA-seq and single-cell high-seq co-assay data set. This data set has over 7,000 cells that are divided across 7 mouse embryo stages and has 1 mouse brain cortex sample. We use chromosome 7 and 11 for testing our model, and we use the rest for training our model. So after pseudo bulking and filtering out all the cell types that had less than 190 cells, we were left with around 10 unique cell types. We held out the last 3 cell types as a way to test the generalizability of our model on samples that originate from the tissues that it never saw or cell type it has never seen in its training phase. Since there are no real existing methods that predict single-cell high-seq from single-cell RNA-seq, so we really don't have a baseline to compare ourselves against. So what we do is we compare ourselves against 4 ablation versions of single-cell GrapHiC. And to test the performance, To test the performance, we used high-C similarity metrics that compared the similarity of the predicted single-cell high-seq contact map with the target single-cell high-seq contact map. We show performance on genome disco, which is a measure of global similarity, usually. High-C-Rep, which measures more of a local similarity. And lastly, we also compare the TADs that we can recover from our predicted map in comparison to the target high-seq contact map to test like how biologically relevant our predicted contact maps are. So first of all, I'll qualitatively show some contact map that came out of our model, and I'll compare them with some of the baseline that we're comparing ourselves against. So broadly, if we find that like all of the baselines and single-cell GrapHiC included are able to recover this global genome organization fairly accurately. But zooming on the region that is highlighted with the box, we see that like these like fine-scale architectural features such as subTADs none of the baselines were able to recover it, essentially highlighting the utility of performing a deconvolution step in our graph encoding part of our architecture. Quantitatively, across the 3 metrics, we find that adding structural support to single-cell RNA-seq was crucial, such as like adding CTCF motif scores gave us like a very, very respectable improvement, 52.7% improvement, which also echoes sort of like the success that previous method had in this space. However, we found that like adding CPG with CTCF was not very useful and did not have a very significant improvement in scores. Lastly, we show that like deconvolving additional structure from the bulk high-C, we can further improve our performance by 5% on genome disco, around 46% on the high-C-Rep metric, and 23% in the TAD similarity measure, highlighting that deconvolution step is really essential in able to recover good quality high-C contact, single-cell high-C contact maps. Next, we test the performance of single-cell GrapHiC on the 3 held-out cell types. And we add this gray dotted line to show the mean score of single-cell GrapHiC on the testing set. And we find that across all of these 3 metrics and all of the 3 cell lines, our scores are very similar to what we observed on the testing set. Moreover, for the exon 1 cell, we did something interesting, which was that we acquired this bulk high-C measurement for mouse brain cortex, and we gave that as an input to our model. And we found that like our model was able to generalize this bulk input as well, giving us a substantial improvement in results as well. Last thing that we test is how single-cell GrapHiC adapts to the structure provided in the bulk high-C. We acquired 5 bulk Hi-C from a study that investigated how the chromatin structure evolves as the cell differentiates from a zygote all the way to eventually form embryonic stem cells. And the key finding was that the structure gets more and more pronounced as the cells differentiate. We, we tested single cell GrapHiC with these bulk inputs, and we found that even when we provided a Hi-C with the least amount of structure, we were still performing better than the single cell RNA-seq plus CTCF baseline, essentially showing that even when we have very little structure available in our input, we are able to capitalize on that. And lastly, just to highlight that all of the results that we've previously shown were on the mouse embryonic stem cells, where we found the where we found the best results. With that, I'd like to conclude by summarizing the contributions of our work. In this talk, I'm presenting single-cell GrapHiC framework, which can predict cell-type-specific single-cell Hi-C contact map from pseudo-bulk single-cell RNA-seq. I have also shown that single-cell GrapHiC can generalize to unseen tissue samples and cell types. And I've also shown, more importantly, that it can generalize to tissue-specific bulk Hi-C contact map to further boost its performance. With that, I'd like to thank my advisor, Ritamra Singh, and our collaborator, Justin Wagner at NEST. I'll also like to acknowledge NIH for financially supporting this project. Lastly, I'll thank the organizers of ISMB for providing me with this very exciting opportunity to present my work here. And thank you.
Maybe one question we can take. All right. If it's a short question.
Test.
Hello.
All right. Did you cross-validate the results using different chromosomes for training and different chromosomes for validation? If not, do you expect any chromosome to be like an outlier?
Yes. So what we originally did, I think these are. I would have to double check. I have these results in my supplementary where we did like a sort of a cross-validation. We put 7 and 11 in the training setup and took 2 other chromosomes out just to see that like our model can still generalize. So, and we found no real change, like no significant change in the, yeah, thank you.
I think it's cool. I'm curious whether you've looked at how the predicted matrices compare to matrices predicted from other kinds. Like, for instance, a matrix predicted from sequence or a matrix predicted by epiphany from epigenomics versus this one predicted from RNA-seq. I mean, obviously you're not competing against them, but qualitatively, like, which ones are better?
So, so it depends. So it depends a lot on how you have pre-processed your target. So it's very hard to do this qualitative comparison. So one thing that we are currently working on is to set up these baselines and see with the same set of pre-processing, with the same set of everything, can we what sort of insights can we draw out from all of the methods that already exist in this space? I would love to talk more about it offline. But yeah.
Thanks so much.